This procedure aims to diagnose melanoma or other types of skin cancer non-invasively using dynamic infrared imaging. Start by taking a white light photograph of the lesion and the surrounding healthy tissue, followed by a reference infrared image of the same region of the skin. Next, cool the lesion and surrounding skin, then remove the cooling stress and record an infrared movie of the reheating process, termed thermal recovery. Finally, perform the data analysis and image processing of the captured white light and infrared images to measure the temperature of the skin as a function of time during the thermal recovery process. Compare the skin temperature at the location of the lesion and far away from the lesion to evaluate differences in the thermal response as an indication of increased metabolic activity that may be related to skin cancer. Ultimately, results can be obtained that show heat generation through dynamic thermal imaging.
Dynamic infrared imaging is a non-invasive, objective, and quantitative method that allows detection of melanoma at an early stage of the disease.
This method can help answer key questions in the field, such as whether a pigmented skin lesion is metabolically active and measure the level of metabolic activity combined with increased blood supply. This can be linked to cancer staging.
This method entails some complexity of acquiring accurate surface temperature data using infrared imaging techniques and compensating for involuntary movement of the subject during the imaging process. So Dr. Muge Pirtini, a postdoc from my lab, and myself will now demonstrate the procedure.
Organize a temperature-controlled exam room equipped with an infrared camera and a PC for infrared image acquisition and storage, as well as a data acquisition card connected to a computer.